Hi everyone! Let's talk about protein synthesis. So you've watched videos about the structure of a cell, about how DNA is replicated when a cell wants to divide. But now we have a functioning cell that needs to go about its everyday business. In order to do that, the cell is going to need to read genes on the DNA. That process of reading those genes and then producing the proteins from those recipes is called protein synthesis. So I'm going to take that entire big process that you watched in the video and boil it down to one simple drawing. So we're talking about reading DNA. So the first thing we need to do is draw a cell, and we need to put DNA in it. Now, where would the DNA be found? DNA is, of course, going to be found in the nucleus. This is our cell membrane. And the slightly viscous fluid inside is called cytosol. And it's in this space called the cytoplasm. So let's put our DNA in here. And DNA looks a bit like a spiral staircase. Remember we said that DNA is like a giant cookbook of protein recipes. Now, when you want to bake a cake, you don't bake every single recipe that's on there. You're just going to bake one recipe. So let's look at our cookbook and pick out the recipe that we want. So imagine that your gene that codes for this protein goes from here to maybe here. So that segment that codes for a specific protein is called a gene. And it's the recipe for a specific protein. When you want to bake a cake, imagine that you have a cookbook in your pantry that has over 10,000 recipes in it. It's massive. It takes up most of the pantry. You don't want to take the entire cookbook out every time you bake a cake. So instead, you're just going to photocopy one recipe out of that cookbook. And then we're going to make that photocopy into a strand of mRNA. The M stands for messenger because it is going to carry the genetic message from the nucleus, from this DNA, out to the cytoplasm where it's going to be read. Now, this recipe, this photocopy, doesn't look exactly like the original. Just like taking a photocopy of a book, the photocopy looks a little different. It's only black and white, maybe the original was color. There's little smudge marks on the photocopy that weren't there on the original. So the same idea here. My mRNA is a photocopy of my gene. So it looks basically the same with a few differences. We have uracil instead of thymine, but it's going to carry the same message. So now this mRNA is smaller than my giant cookbook, and it can leave through little pores in my nuclear membrane and go out into the cytoplasm to a structure that's going to produce that protein. And if you remember back to cell structures, you have kind of a two-piece structure here made out of RNA that is going to act like the chef. So this is our rRNA, and the little r here stands for ribosomal. So that's my ribosome. And he's also the chef. So he's going to read the recipe and actually build my protein. Now I need him to read this mRNA, so I'm going to feed that mRNA through the ribosome like this. And the ribosome is going to read this mRNA in 3-letter blocks, or 3-letter words called codons. And it's going to spit out a protein out the top. Every 3 letters tell it to grab an amino acid. And then the next 3 letters tell it to grab another amino acid. The next 3 letters tell it to grab another amino acid. And so on, until I have formed a polypeptide chain. And that polypeptide chain will coil up and be my final protein. Now we're missing one thing here. Once my chef is busy reading the recipe and baking the cake, or in this case, building a protein, he needs somebody else to bring him the ingredients. So we need one more type of RNA, and we're going to use a molecule called tRNA. The T stands for transfer, because he's going to transfer the correct amino acid to the ribosome. So he's kind of like your sous chef or your assistant chef. And he's going to bring the correct amino acid to the ribosome. Now, tRNA molecules are kind of dumb as a box of rocks. So, this tRNA molecule might be holding this amino acid. So imagine that I am a chef, I'm a ribosome, and I yell out that I need a cup of flour. And a sous chef comes running over and he says, "Here you go, here's your cup of flour." And I, as the chef, say, "Great, could you also go get me a pound of butter?" And my sous chef is going to say, "Look, dude, I don't do butter, but if you need more flour, you'll let me know." So that means if there are 20 essential amino acids, you need to have 20 different types of tRNA molecules that can each bring their own specific ingredients. Now, what I like about this analogy of kind of being a chef and baking something from a recipe is it helps remind you that nucleic acids, like DNA and RNA, are different types of molecules than proteins. Proteins are made out of amino acids, and all of these nucleic acids are made out of nucleotides. The chef doesn't crumple up part of the recipe and throw it in the cake mix. He doesn't cut off his fingers and put that in the cake mix. That'd be kind of gross. So these are separate types of molecules. The protein is what I'm building. The nucleic acids help to build those. Also, just like this analogy, if I have a recipe, I can read the recipe more than once and bake more than one cake. So a ribosome can read this mRNA strand multiple times and make multiple copies of the protein. In addition, another ribosome could attach and start building its own proteins using the same recipe. Now, over time, eventually, my mRNA is going to get crumpled and it's going to get ripped and it's going to get smudged, and I'm not going to be able to read it properly anymore. If that's the case, then I have to go back to my original cookbook and make a new photocopy of that recipe. Now, this process that we just broke down here is called protein synthesis. And there are two main steps to protein synthesis. And they are what happens in the nucleus. So where I'm photocopying the gene onto mRNA, that is called transcription. And where I'm actually reading the recipe and building the protein, that is called translation. So transcription happens in the nucleus and translation happens in the cytoplasm. And the way you can keep the two straight is think about the words. A scribe is somebody who writes things down. So for example, Stephen King, who's an author of books, is a scribe. If he wants to sell his book in another country, let's say China, before he can translate the book into Chinese, he has to first write the book. So you have to scribe before you can translate it into another language. All right, and that's your quick overview of protein synthesis. Now, one more quick thing I want to talk about is when we're doing this process of photocopying, or even when a cell is dividing and it's splitting the DNA and making copies during DNA replication, it is possible that I make a mistake and sometimes put the wrong nucleotide in there. When that happens, I can end up with a mutation. So a mutation is basically a mistake in the DNA. If I accumulate enough mutations, I can eventually end up breaking the cell in a sense, and it starts to divide out of control. Now, there are two types of genes that really help to regulate how often a cell divides. There are oncogenes. So oncogenes are really the genes that regulate cell division and tell a cell when to divide. And tumor suppressor genes limit cell division. If I have a mutation in one of these two genes, then maybe I don't limit mitosis and I start to divide out of control. Or maybe I tell the cell to divide more often than it should, and it divides out of control. If it divides out of control but stays localized, then we say that it is benign. But if it moves to another tissue from where it originated, then we say that it is malignant or it has metastasized. At this point, we consider that to be a cancerous cell. So for example, my father passed away in December 2019 from pancreatic cancer. Now, what really took him down were the cells that had moved into his liver and formed tumors there. Now, when he was first diagnosed, they saw these masses on an MRI, and they can't really easily get to the pancreas because your pancreas is kind of tucked up underneath your stomach. So it's not easy to get in there, pull some cells out to take a look at them. But the liver is this nice big organ over on the right side in that upper right quadrant. So they took a biopsy of his liver cells, specifically the ones that were in these masses that had formed. And what they found when they looked at those cells were markers on the cells that are markers you would find on a pancreatic cell. So that's how they know that the cancer that he had in the liver is really pancreatic cells or pancreatic cancer as opposed to liver cancer. So we tend to name a cancer by where it originated or where those cells came from. Now, you might have always wondered, why do you call somebody who studies cancer an oncologist? And now you know why. Because they're studying oncogenes. They're studying those genes that regulate cell division. Not to get too far down the rabbit hole on this one, but if we just take a quick look and ignore all this text here, If we have healthy cells and we start to either get oncogenes turned on when they shouldn't or suppressor genes turned off when they shouldn't, then we start to get cell division happening when it shouldn't. What happens then is that those cancer cells actually induce the formation of blood vessels. So it causes blood vessels to move into these tumors, really nourishing these cells that are dividing out of control. And in addition to nourishing them so they can keep dividing, it also gives them access to the roadway that goes through your body. So now those cells, you can see here, can migrate into those blood vessels and travel to other parts of the body. All right, I think we're going to leave off there. And if you have any questions about cells, you can put that up on the discussion board.